open ಆಗ್ಲಿಲ್ಲ.
Oh, okay.
So we have this launch Zoom and it's not going away.
No, it's nothing to do with that.
Stop sharing.
Yes, we now can see it.
Okay, that's good.
All right, we're good.
Excellent. Okay, let's get started. So thank you, Pushpa, for this opportunity to share about COVID-19 vaccine situation in the world. So I work at Ohio State and now involved in COVID-19 research. I thought I can share few of the updates on this situation. So before we start something, always you should thank the people who started the vaccine research and especially developed the vaccines way back in 1800 and 1900, which saved millions of lives. So this is the beginning of the vaccines, which today we are dreaming to come for COVID-19. So Edward Jenner and Louis Pasteur are the world well-known names. So why you need vaccines? So if you look at the first curve, if you have any people initial exposure to any virus or a vaccine, this is how the primary immune response induced in people. So this takes typically between 2 to 4 weeks to get this kind of response. However, if you have a second exposure to a virus or any pathogen or a booster vaccine, the response will be faster and it is to a many-fold higher. And as a result, person won't suffer from the disease and it helps to fight against the infection. So just to introduce about COVID-19. So COVID-19 is a huge pandemic of 100 years. So this is because we had a huge outbreak of Spanish flu in 1918. That Spanish flu infected 1/3 of the global population and killed 50 million people throughout the world. And 675,000 people in the US died at that time. And afterwards, there were 8 flu pandemics happened. But still, those other pandemics did not have such a huge impact. And this COVID-19, in March 2020, WHO declared it as a pandemic. Since then, it has spread to almost all the countries in the world. So what lessons we learned from the previous pandemics is generally after any pandemic, it takes around 1.5 to 2 years to build the herd immunity. So what do you mean by herd immunity? That means people have the immunity against the disease for reinfection. So to achieve this herd immunity, so we need now 60 to 70% of the people should become immune either by exposing to infection or through vaccination. So then only we can say we are safe from future outbreaks. So this is very important because like flu, coronavirus, the COVID-19 is also an RNA virus. It genetically goes through so much changes every cycle. So every cycle of replication of the virus, which leads so many mutations, and all the mutations are not harmful or lethal to humans. Even 1 in 1 billion mutation may be highly lethal. So earlier pandemics have given the information that after the initial outbreak, there are 2 to 3 spikes, so which resulted even more deaths. So what we learned about the coronaviruses. So before COVID-19, we had 2 coronavirus infections. One is SARS, it is severe acute respiratory syndrome, and which happened in 2003, which infected close to 80,000 people, and 10% of the people died out of SARS. And later in 2014-15, we had MERS. It is Middle Eastern Respiratory Syndrome, is also a coronavirus, which was more lethal, which killed more than 30% of the infected people. However, these 2 viruses were not highly infectious like COVID-19. Those 2 viruses, they infected only the lungs, and the transmission was only by direct contact. Unlike COVID-19, droplet infection is involved, and the upper respiratory tract infection, that is throat infection, is involved. And this virus is highly infectious. That means even if maybe 100 to 200 virus particle from a single droplet, which can have so much of virus, can cause disease. So COVID-19 is a huge monster, so that everyone definitely feel that. This virus, what I'm showing is the electron microscope picture on the top. The size of this virus is around 100 nanometers. So just to give in a common language, if you take a mustard seed and make 100,000 pieces of it. So you imagine that is the size of this virus. And schematically, if you see the viral structure below, there are spikes on the virus surface. This is what people have been using to make vaccines. However, the duration of immunity to COVID-19, either infection or vaccination, is not known now. Even though people are infected and recovered, or even if you find a vaccine near future, how long that vaccine works, we do not know. The reasons are earlier respiratory infections, immunity to vaccine or infection to recovery did not give any long-lasting immunity. It varied between 6 months to 1 year, not more than that. So a good example is the flu. So what are the different types of COVID-19 vaccines which are under development at the moment? Are inactivated virus, that means grow large amount of virus and then kill the virus and use with some adjuvant by intramuscular injection. The other one is take the viral fragments, not the whole virus, like the spike protein, which is predominantly used now as the vaccine antigen. And then people now, the most popular one is the mRNA vaccine, is a fragment of the viral RNA, like the spike protein RNA. So because it is very easy to manufacture large amount in a very short time, that's why the mRNA vaccine is, people are trying that. And the vector vaccines, you would have heard about this adenovirus vaccines and measles virus recombinant vaccines, which now in UK, they are trying to manufacture. That I will tell a little bit detail in the next slide. And then the technology which is widely used is the nanotechnology based vaccines. So why I'm introducing all these vaccines, different things are each vaccine formulation have got their own advantages and disadvantages. And the earlier studies with the SARS and MERS vaccine studies people performed at that time, they found that some of the vaccines cause more disease. So that's why people are very careful now to introduce which one is more safe. And globally, at the moment, there are close to 102 plus COVID-19 vaccines are in development. And 8 of them have already been approved for clinical trials. So what do you mean by that? So to develop any vaccine and to answer why the COVID-19 vaccines development is getting delayed so much, is it follows all these steps before the vaccine comes to the market. So initially they have to find out the pathogen and find what vaccine platform is suitable for this particular virus. And then they have to test in animals, that's called as the preclinical studies. And then it goes to human studies. Like there are 3 different phases, 1, 2, and 3. So phase 1, they have to check the safety involving some 100 plus people. Phase 2, some 1,000 people. And if it comes to phase 3, where they check for the efficacy, wherein there are several thousand people are involved. So each of these phases takes at least 6 months to 1 year, sometimes even more. And then it goes to regulatory like FDA and CDC approvals to see how FDA can investigate the safety of these vaccines. Then it will go to manufacturing large amounts. Then people also find quality by checking the vaccine formulations. And even after 1 year of vaccination, are the people not having any side effects? Why I'm telling all this is, even after they release any vaccine, even 1 in 1 million people, if they have any side effects, which leads to lethality, you know that it becomes viral and people start hating the vaccine and the vaccine companies will be sued. And nobody wants to have that kind of reputation and lose their whole business. And nobody keep the vaccine ready for any pandemic disease, mainly because the earlier MERS and SARS viral vaccines, what people have already developed, though they are not commercialized, they don't work against this COVID-19. Okay, coming to the most important slide here now. So there are, what are the promising vaccines at the moment they're available for COVID-19? The first one is in the US, is a mRNA-based vaccine. This is a company called Moderna and NIH have a collaboration. For this vaccine, US government has invested $483 million and it is already in phase 1 trial. So though there are no mRNA vaccines at the moment in the world are used for any disease. And this vaccine has not been proved earlier, even in other animal models. And straight away it come to the phase 1 because for the Zika virus, a similar platform has already been tested. So based on the promise which had, and it can induce a good immunity, and it is within couple of months, they can make millions of doses. So because of that promise, now the US government is going in war footing to develop a vaccine before the end of this year, so to help just further spread. That is in the US. And in UK, this is called the chimpanzee-derived adenovirus vaccine. This is adenovirus is a DNA virus. They have this viral spike protein inserted into this adenovirus. And for this, UK government has invested. And most importantly, Serum Institute of India, now even before this phase 1 studies results are out, already the manufacture has started. Usually that won't happen because people wait before they invest money on this. But Serum Institute of India is the biggest vaccine manufacturer in the world, and now you volunteered to make 40 million doses of this vaccine by October 2020. They are hopeful that this phase 1 trial will give a positive result, so people start getting the vaccine. And in China, there are already 3-4 vaccines are in development. They are in phase 2 trials. People don't trust more about that product. We want to depend on the top 2. And in Germany and Switzerland, again, there are many mRNA vaccines are in various stages of development. The problem is, what is the safety of these vaccines? And how long these vaccines, whatever they come up, they are going to give protection? So these are the big questions. Still, people are not very clear. So because it takes time, and based on the earlier vaccines, generally, they took between 5 to 15 years to come up with a good vaccine to protect the people. And especially for the RNA viruses, the prior studies, for example, for flu virus, you must be aware of it. Every year, close to 70 to 80,000 people die in the US because of flu. Instead of 30 to 40% of the people, they get vaccine for flu. And they update the vaccine virus every year. Instead of that, the protection is not complete. So, and the acceptance rate is also, as I said, is less than 50%. So that's also an RNA virus. So we do not know in case of COVID-19 how the situation will be, because we are also scared of the spikes. That means once the lockdown reopens, when the people start getting into moving around, and the new genetically variant virus, if it comes out, there are chances even the previously recovered people may also get infected. There are chances are there, because earlier pandemics have the history of a couple of spikes up to 2 years before they came under control. However, now the scientific community is working really hard. I'll tell you, there are funding opportunities for COVID-19 so much for people to explore their different vaccine platforms, because we do not know which one will give the best possible immunity. So that one will be used mostly, because there will not be one magic bullet for this viral infection. So we need to have multiple options. And it is not that who is going to win the race. It is the first priority is we need to have something at the earliest possible. And even if it is a 30, 40, 50% protection, like how we have for a treatment, some drugs like remdesivir, even if it is 30, 40% protection, still we are hopeful it is going to help us. Similarly, we need to have a vaccine, and then to come to know how those vaccines perform, we need to wait. We need to see the responses. So in addition, as you know, for the response of the vaccines is not the same in every individual. It varies with the sex, age, and then the environment and the pre-existing immune conditions. And so many factors are involved in response to a vaccine when we go for mass vaccination. Anyway, so I think these are all the most important points I wanted to cover in 10-15 minutes, because I wanted to leave more time for discussion. And fortunately, in Ohio State University, we have a facility, it's called a BSL-3 facility, to handle this kind of viruses inside this. This is a special facility which costed around $23 million, which is in our campus, to use viruses like COVID-19 inside this facility. We have already started going for training, and they're applying for grants to use the facility people already have grants at Ohio State, including my lab, to test the proof of concepts. The studies are in progress. The scientific community is hopeful and promising compared to earlier pandemics, because this has become a big challenge to all the researchers in the world. So I hope I conveyed some important messages, and I'm happy to take questions to answer what you have in terms of COVID-19 vaccines. So thank you.
Thank you so much. It was really an honor to hear such a credible source of information, so much information of what is going on. I know everybody's curious and everybody's hopeful for the beginning of the end. And thank you so much for sharing all of this information with us.
Thank you.
ಕ್ವಶನ್ ಕೇಳ್ಬೋದಾ?
ಖಂಡಿತ.
ಆರಾಧ್ಯ ಅವರೇ ನಮಸ್ಕಾರ.
ನಮಸ್ಕಾರ ಗೋಪಾಲ್.
It was amazing. It was a pleasure to hear your excellent scientific analysis, and bala santosh aitu. You did a great job. I had a question about your personal thoughts on which of the methods you think are actually can be brought to market with the greatest efficacy and most safety. And the second question was, will you be doing animal trials of companies based on the work that you're doing?
Yeah, good question, Gopal. It's very relevant. I did not talk all those things. I wanted to answer to the question. Okay, so the first question is about what vaccine is safe and then more efficacious, right? So the vaccines, as I mentioned before, there are different types of vaccines are trying, and many researchers are worried the vaccines which are given by injections, they won't provide a good cross protection if a different virus strain comes up after a few months. And also it may not induce more of mucosal immunity in the respiratory tract mainly, because where the virus principally replicates. A good safe vaccine you should come up with a vaccine which can be delivered by intranasal, like a flu mist or something. And then you should have a multiple viral antigens, like it not be spike alone, because viruses, when they genetically change, the spike protein is the one which genetically prone for change. So that virus vaccine may not work if the virus comes with the mutation later. So you should better to not only target the antibody-based immunity, what currently many vaccines they are trying, you should also target the T-cell immunity that has a long-lasting memory response. That's what people and we are also proposing from here, include the viral nucleocapsid protein antigens so that we can induce a good T-cell immunity for the conserved viral regions, so we can come up with a vaccine if we have a good studies in the animals. So coming back to animal trials, so at the moment, ferrets, hamsters are readily infectable by the COVID-19 virus. And especially in the ferret model, the aged ferrets, they are also dying out of COVID-19 like the 70 to 80 years old people when they get infected to COVID-19. So people are proposing to use the suitable animal model where it mimics the viral infection, and also use the rhesus macaques. So as you know, rhesus macaques, the problem is the availability. And the second thing is, as you know, more than 80 to 90% of the people, they may not have a disease. So we'll have a similar situation even in macaques when you're doing the trials. So these are the models people are using. So did I answer your questions, Gopal?
Yes, you answered the question, but again, I'm sorry, I'm taking up a couple of questions. I hope you don't mind.
Sure.
The first is, I thought pigs were closer to humans for many trials, and I was surprised not to see pigs. And number two, I'm also asking whether you have heard of the fact that Iran had so many deaths while Iraq did not have that many deaths. And people all over the world have found out that some countries have more severe response than others. It is not related to density of population. It is something else that's going on. And I wonder whether you had some thought about genetics of the human body and the response.
Okay, I'll come to that. So one first thing is about the pig model. Yes, I agree. In fact, my lab works more on the pig models, and the respiratory system is very similar to the human system for the responses. However, this COVID-19 virus doesn't infect the pigs at the moment. So we need to come up with the repeated passage in the pigs may start infecting the pigs, but it is not readily now infecting the pigs, this virus. So we have the models like the porcine respiratory coronavirus, which is a native of the coronavirus, which also causes the respiratory infection. So people won't trust, the granting agencies won't like to see something we are going to develop starting from making the virus to infect. It's better to choose a model which infects readily and come up with a vaccine. And then coming back to why there are differences in the mortality and how the disease is causing the scare throughout the world, like Iran, even in India. So we are all worried that in India, people, if it happens like in the US, the people die in millions, right? So the reasons are several. So one, people are coming up with the strategy, the explanation that the viruses which are infecting the population in different countries is not the same. There are some genetic changes in the virus. Still we need the investigation and the precise information why there are big differences. And there are some publications already saying in India, they have a microRNA, that is an innate microRNA, which can inactivate the virus, which is a inbuilt immunity. And then the other thing is, not only this coronavirus, there are 4 or 5 other human coronaviruses which are circulating in the human population. If the immunity to that virus is prevalent in those countries, the susceptibility to this COVID-19 will be minimal. So like this, there are multiple things maybe helping in other countries, especially the developing countries, Asian countries, which are less prone for the infection compared to the Western. Make sense, Gopal?
Yes, thank you very much. That is wonderful. Okay. No more questions.
Okay. So, Aradhya, tumba chanagittu. Very good overview.
Thank you.
I'd just like to remind the viewers that today's edition of New York Times has a collection of 5 or 6 articles about the vaccines in relevant to COVID-19, including an article that BCG is being investigated, oral polio is being investigated as a tools to reactivate innate immunity, not specific immunity, and the politics of vaccines, the cost of vaccines, the time frame of vaccines, the entire spectrum of issues surrounding the vaccine trials for COVID-19 are all available. And the COVID-19 articles on New York Times are all free. So they're not only for members. So anybody that goes to today's issue of New York Times can find all these articles. They're extremely informative.
Thank you, Naveen.
I have a question. Thanks, Naveen, for that information about New York Times, and thank you, Aradhya, for the information. ನನ್ನ question ಏನಂದ್ರೆ, is there a permanent solution for this? ಯಾಕೆ ಹೇಳ್ತಾ ಇದೀನಿ ಅಂದ್ರೆ, okay, we develop a vaccine, we spend so much time and effort, and we develop a vaccine, and that vaccine, like, it's probably for a year. And iದೇನಾಗ್ತದೆ ಅಂದ್ರೆ, every 3 to 5 years, China is manufacturing something new. So let's say 3 years from now, we are in the same state. COVID-19 ಬದಲು COVID-22 or 23 ಬರುತ್ತೆ. ಅವಾಗ ಏನಾಗುತ್ತೆ? I mean, are we planning for the future? If something happens, can we turn around and act fast in 1 or 2 months? Like, how it's going to work in the future?
Okay. My answer for this, based on the vaccine area, what I've been involved to flu viruses. If for information, 1918, there's a pandemic for flu happened, right? 100 years over. Even today, we have a vaccine. If tomorrow, if we get another flu pandemic, which can completely protect. Even we are not successful in protecting against the seasonal flu completely. This is because the vaccine platforms, what people are in marketing, are not highly efficacious. So they are worried too much about the safety, and then because it's also important because it has to cover the different age population, people with different health conditions, and that is the one reason. If you come to the COVID-19 kind of situation, compared to flu, I feel controlling a coronavirus is a lot easier, mainly because we do not have so many viruses like the flu viruses which are coming out. So we had a history now of the SARS in 2003 and 2015 MERS and 2020 COVID-19. So I'm sure with the available opportunity and how the scientific community is going with so much of investment in their thoughts and the techniques, they'll come up definitely with the vaccine which has a broader immunity. That's what I mentioned, including more of the conserved viral regions in the vaccine formulations, so that people will have the memory for longer duration and they won't suffer like how they are suffering at the moment. So your point is absolutely right. So if you have that kind of vaccine already available, it is very easy to go and manufacture large amount in no time without spending time on different, I told you the steps involved in making the vaccine for marketing.
So are you saying that, say maybe in 3 years from now, on Vasa virus bantandre, we don't have to scramble like we are doing now, running pillar to post. We can turn around something in a matter of couple of months, new vaccine?
Exactly. So if the manufacturing part is only the part is involved, and then if the people, because what happens if there is no coronavirus, if there is no infections next year, people will not take vaccine, right? Mm-hmm. So at least if the technology is available, if they know it covers the increased breadth of immunity against all the coronaviruses by making a vaccine which is broadly reactive, including not only the spike protein which induce only the antibody-based immunity mostly, and including the vaccines which can induce good memory T-cell immunity, so which is relatively long-lasting. So there are very many ways to do that. That's why now, if you I'll tell you, NIH has put in so much of money for researchers to come up with any vaccine which can give a long-lasting immunity. But to test that, it takes several years, right? So they're investing money. I think now the whole world community is looking up the developed countries which have the resources and also suffering more to come up with something to help even the developing countries.
Thank you so much. I appreciate it.
Thank you.
Last question, no?
Sure.
Was that the last question? Thank you so much, Doctor. This was very, very informative, and this discussion was even more helpful and informative. So thank you so much for joining us today.
Thank you. Oh, thank you, Mahesh.
What information?
Thank you, Aradhya.
Oh, thank
presentation ಚೆನ್ನಾಗಿತ್ತು. ಒಳ್ಳೆ information vaccination ಬಗ್ಗೆ.
Thank you, sir.
ಇನ್ನು ಯಾರಾದರೂ ಮುಂದೆ ಫ್ಯೂಚರ್ ಅಲ್ಲಿ ಡೌಟ್ ಬಂದ್ರೆ ಐ ವಿಲ್ ಟ್ರೈ ಟು ಆನ್ಸರ್. I can find out for you. And we are all in the bad time that nobody expected. After December, we saw this virus coming out. It's going to cause so much of fear in people. But still, we are safe, and we hope things will be better.
Yes, sir. Thank you.
Thank you.
Mental well-being is the precursor to physical health. Nijatane. So, ee thara situation iravaga, it is even more important that we are mentally happy and relaxed. So, please join me in welcoming Shri Shyamasundar Tirumali, live from Mysuru. So, ಅವರು ನಮಗಾಗಿ ಕೆಲವೊಂದು ಹಾಡುಗಳನ್ನು ಹಾಡ್ತಾರೆ. So, please, thank you so much for joining us today.
Okay, correct.
ಇನ್ನ ಆನ್ಲೈನ್ ಬಂದಿಲ್ಲ ಅನ್ಸುತ್ತೆ.
ಇನ್ನು ಮ್ಯೂಟ್ ಅಲ್ಲಿ ಇದೆ ಒಂದು. He knows Naveen. He knows. ಇಲ್ಲಿ ಇದ್ನಲ್ಲ ನವೀನ್. ಏನು?
ಡಿಸ್ಕನೆಕ್ಟ್ ಆಗೋಗಿದೆ.
ಇಲ್ಲ ಅವ್ರಿಲ್ಲ, ಡಿಸ್ಕನೆಕ್ಟ್ ಆಗೋಗಿದೆ ಅನ್ಸುತ್ತೆ.
ಹೌದಾ? ಒಂದ್ ನಿಮಿಷ. ಅಷ್ಟೊತ್ತಿಗೆ ಬೇರೆ ಇನ್ನೇನಾರು ಮಾತಾಡ್ತಾ ಇದ್ರೆ, ಇನ್ನೇನಾರು ಕ್ವಶನ್ಸ್ ಇದ್ರೆ.
Anyway, until then, I'll be speaking generally about this COVID-19. So if you guys also have the questions, we can just have a informal chat, right?
Aradhya,
yes.
One question. This is Yunus.
Yunus.
The talk was very good, very informative, especially on the microRNA. I was not aware of that. One thing at our institute, right, at University of Maryland, I just wrote a proposal for COVID-19 for our internal grant. Okay? So we are a structural biology institute. So one thing we found, we figured that including the nucleocapsid protein in the vaccine, right, it's been associated with severe allergic cytokine storm and so on. Okay, so we decided to exclude that from the antigen design. Okay, so we just went. We are now going with the membrane protein and the spike. Okay, and we have some mosaic antigen design. I mean, that's our specialty. So that's the only thing. But it's surprising that the pigs are not getting infected, considering the fact that if you take the receptor binding domain of the spike protein with the ACE2, right? So there is only one amino acid change there, difference between the key binding residues. But anyway, we are also going for the ferret model. Okay, we don't have pigs, of course.
Yunus, thank you. To introduce, his name is Abdul Yunus. He was my classmate in the vet school, and he works in University of Maryland. And he was a very highly talented person. He's involved in
thank you.
The intellectual protection and patent related studies in University of Maryland. So he's also PhD from University of Maryland, and he's doing extremely good work on the structural biology. So thanks, Yunus, for sharing. It's a good information for vaccine developers.
Thank you. Thank you.
Hi, Aradhya. This is Murali from Omaha.
Hi, Murali.
How are you?
Good, good.
Hey, I have a question for you.
Sure.
I know that data is very limited, but I was wondering whether it's cellular immunity or antibody response, which is important in protection against this coronavirus or COVID specifically.
So based on 2-3 publications I read before, the younger people, they have a very weak antibody response, but better immunity. So they're associating this mostly to cellular immunity, cell-based immunity. For people who are aged more than 50, they found high neutralizing antibody titers. So in the infected people. So that and then also they recovered. So they associated that to antibody-based immunity. So that's why in humans, so depending on the age, some people are protected by cell-based immunity predominantly, and some for antibody-based immunity. That's why finding the antigens which can induce both, like how Yunus mentioned about the matrix protein, it may be a good choice. And spike protein, of course, if it's required. This also has got the T-cell epitopes. So like this, we need to have a vaccine which can induce both antibody and cell-based immunity, and long-lasting memory is a dream for us. Is that answered your question, Murali?
Yeah, thank you. Yeah, it's going to be challenging, I guess. Not to scare other people, but
yeah. But now the way how the people are now going back to lifting the lockdown, and still the number of deaths you are seeing close to 2,000 in the US and other countries, we do not know what we can expect down the line in another 4-5 months. It may not be seen immediately tomorrow. So we see the impact only after a month, right? After a month, you will definitely see. Now I heard in India, especially in Karnataka, so they're all lockdowns lifted. People are back to work and there has to go, though the public transportations are not started, but do not know what kind of consequences you will have in another couple of months, right? You should cross our fingers and hope we will not have more problems.
Yeah. Antibody test kits, I think that can rule out. I mean, that will be a big help. I'm sure a lot of people are exposed, but I don't know what percent of the population shows asymptomatic or develops asymptomatic disease. I think it's very critical to know the antibody status or immune status.
But
the population.
The problem is just for the detecting antibody positive or negative is not going to say whether the person is protected, mainly because it may be false positive. It may be reacting to other human coronaviruses.
Yeah.
So it's very difficult to come up with an antibody-based assay to say precisely it is because of the COVID-19. unless and until you come up with the specific non-cross-reacting, they highly focused antigen and a solid assay, which is it can be done